hear me, not just the people in the room. You've all taken your cameras off, so I can't see you. Suresh, can you hear me?
Yes, I can.
Awesome. We can hear you too. Welcome to our New Zealand EV Journal Club. Well, we decided that we'd do something a little bit different this week. What happened over lockdown was, as many of you all know, is we decided to set up a weekly journal club. And this has been really popular, and we've had people attending from Wellington, Christchurch, Dunedin, Auckland, Palmerston North, and we've mostly to date been presenting journal articles. But what we thought was, in lieu of our heavy symposium, which would normally run on in July, we thought we'd start inviting our international speakers. And unfortunately, Suresh has walked down in Melbourne again, I hear. Sorry, Suresh. We had hoped to invite him over, but instead we've got him on Zoom today. So I also have to, I've got my notes here, I have to thank Anastasia for organising the journal club so well over the lockdown period as well. So thank you, Anastasia. Professor Suresh Mathivanan is from the Trobe University in Melbourne. We met probably guessing, and he is the equivalent of Larry. Thank you. Oh, there we go. Anastasia's on the mutant people. He is the director of the Australasian EV Research Centre at La Trobe University. I would say that Suresh is very much a jack of all trades. He's a little bit like me, where we have dabbled in vesicles from everything you can possibly think of, from milk and fungi, and today he'll be talking about cancer vesicles. His background is in proteomics and bioinformatics, and he's developed some incredible tools. So I Googled you, Suresh, and I found that you had 19,812 citations for your papers. And a lot of that is due to the development of an amazing proteomics analysis tool called Funrich, and a couple of vesicle databases, Vesiclepedia and Exocarta. So we are absolutely delighted to have you with us virtually, Suresh. next time, I promise we'll bring you a visit. So without any further ado, I'll hand over to Suresh. Thank you very much.
Yeah, thank you, Sherry, and thanks to the organizers for the invite. I should start saying that I'm pretty much jealous seeing you guys pretty much in workplace. I can see a lot of you guys maintaining social distance and still in workplace, but we are in second lockdown now. Recently, it's been almost like 80 cases in Melbourne. So yeah, I haven't seen my office for almost 3 months now, and I'm pretty jealous. What do you guys have achieved? Okay, so what Sherry said, so again, I'm from the Research Center for Extracellular Vesicles, and definitely we should have a really good tie between New Zealand and Australia in terms of the two centers. And just to give an overview, I'm sure all of you know about the center that we have in La Trobe. Again, we call it RCEV, the Research Center for Extracellular Vesicles. And we have somewhere like 10 groups who are specifically focused on various topics of extracellular vesicles. As Sherry mentioned, some of you really know a lot of these researchers. For example, Andy Hill, whom you know really well, and he works on neurodegenerative diseases. Merlin Anderson, again, on fungal and plant EVs. You have David Greening, who works on reproductive biology and EVs concerning to that. Mark Hewlett and Ivan Poon on apoptosis and cell clearance-based EVs. And Maria on bacterial EVs. Similarly, me and Richard on cancer and many other things, and also Jackie Orion. So we do have these 10 researchers, and we also have a small journal club going through, but we do have it once a month, not as probably frequent like how you guys have. So as a lab, as Sherry mentioned, kind of I work on everything. Whoever comes to me, I kind of work with them, and my head of the department calls me a prostitute because of that. I've never said no to anyone when they come to me for working together. But off late, there's too many things on board, so we kind of focus on few things, and these are the things which we focus more on. In a way, EVs in metastasis and cancer cachexia, in intercellular communication, cross-species communication. We do a lot of work on identifying what are the molecules which regulate EV biogenesis. And also we use proteomics wherever we can, and any study that we do, we always complement that with proteomics, and also bioinformatics, which is also our strength. So I just have to reiterate here, because as we all know, the field of extracellular vesicles, the terminology is pretty much a soup. And we often get confused with what to use. We normally call these vesicles extracellular vesicles, but still, when I try to pitch a particular paper to a journal, a bigger journal, then I use the term exosomes, because it's more of a shopping terminology that you can sell easily or advertise easily. But I just have to reiterate that the vesicles that we are working with is not pure in any certain population. It's probably as heterogeneous as anyone can isolate. And even though I use this terminologies, exosomes and extracellular vesicles, mixed in this particular talk, you can consider these are extracellular vesicles, and it's not for isolated to homogeneity, right? So in a way, that's the crux here. So again, 2007, we do know about this paper, and that's why I'm reiterating this particular point, is because we do know that extracellular vesicles mediate intercellular communication. And this paper by Jan Lotwall kind of triggered me to getting into this field, because 2007 is when I started getting into this EV field, and that's predominantly because of this paper. When I studied or when I read this, and I got interested, saying that how RNAs can be transferred between cells, and then these RNAs can be translated into proteins, and then they can be functional on the recipient cell. In the context of cancer, there are so many articles now. I'm sure all of you know, and the literature is full of many articles. But I would like to highlight only these two articles from David Leiden's group, because not only because of their influence in the field, when you talk about cancer, and most of the times when you talk about non-EV people, you would like to mention some high-impact journal which has the potential of good reproducibility. And this particular article has been reproduced by many groups, and even the international reproducibility group, where they try to reproduce some of the important findings of this. We're able to reproduce everything in this particular study except for the use of c-Met, where they were not able to prove that c-Met was the one which was kind of driving this phenotype, but they were able to reproduce that EVs can create pre-metastatic niche. Not only them, but many other groups were able to reproduce this particular finding. Similarly, David Leiden's group were able to show that EVs drive organotropism, and where it was unclear as how certain cancers can spread to lung, liver, while some other cancers can spread to brain and the other organs. And these guys came up with these EV-based signature. They kind of proposed integrins, which are present on EV membranes, could be the driving factor for where the tropism can occur. Right? And this study is also pretty well regarded in this field. And again, based on this, we do know that EVs have a lot of role in cancer, and we kind of have been working on cancer for some time. And I'm going to present two small studies which have been published last year, end of last year to be precise. So again, we do know that tumor microenvironment is not by the cancer cells itself, and we do have various other cells, the stromal cells, the immune cells, including the cancer-associated fibroblast. You do have the endothelial cells. We do have the immune cells in this vicinity as well. All of them collectively interact together, and then thereby they can aid in cancer progression. And the role of extracellular vesicles in this particular process, including facilitation of angiogenesis, proliferation, evasion of the immune system, epithelial mesenchymal transition, metastasis, and also evading apoptosis, has been well documented in the literature, some more than the others. But we do have this kind of study in the lab where we look at the cancer cell and we look at the bidirectional talk of the cancer cell with other cells in the tumor microenvironment in the context of EVs, right? So even though we study their role in terms of what pathways that EV can activate in the stromal and the immune cells, and what message would you get back from those endothelial cells when they're challenged with and without EVs, right? So even though we do study in those contexts, but in this today's talk, I'm only going to focus on cancer cell EVs talking to other cancer cells in the tumor microenvironment. So again, we do know that there is a lot of heterogeneity in the tumor. There's a lot of intratumor heterogeneity within the clonal populations. So what I mean by that is we do have various mutational loads of the cancer cells. There are some which would say, for example, they may have P53 mutation, and there are some which would have KRAS mutation, and there are some which have both these mutations in them, and some which are normal cells in this context. And how would exosomes or extracellular vesicles kind of communicate with these cells, and how will they transfer the aggressive phenotype, or in a way, how will they favor cancer progression in this context is what we are going to study, right? So in this first first part of the talk, I'm just going to focus on neuroblastoma, and this was predominantly done by the PhD student Pameli Fonseca. Now she's postdocing in my lab since last year, where she graduated. And this is during a PhD where she developed this particular study. So where we looked at the role of extracellular vesicles in aggressive neuroblastoma cells and how they can transfer the aggressive phenotype from one cell to another sensitive cell and render them more resistant to treatment, as well as transfer the phenotype more into a more aggressive phenotype, right? So neuroblastoma, a little bit basics on that. Again, it's a pediatric cancer. It's of the sympathetic nervous system. It's responsible for at least 15% of childhood cancer mortality. What is most striking about this particular cancer is it's very aggressive, and it's kind of one of the cancers where the treatment doesn't benefit most of the patients, especially those with high-risk neuroblastoma. Right. So when you talk about high-risk neuroblastoma, one of the characteristics of high-risk neuroblastoma patients is this amplification of this transcription factor called NMIC, which is normally found in at least 20% of neuroblastoma patients. It directly correlates with tumor aggression, also treatment resistance in a way. Because we do have c-MYC overexpression in the other subset of the patients, but in these 20%, you have amplification of NMIC, and that's why it's kind of considered high risk. In most of these patients, 20% of them, there's at least more than 10 copies of NMIC in them, suggesting that you will have a lot of protein being expressed in these cells, and these transcription factors kind of go pretty crazy in those cells, and that's why we see this phenotype. So what we did in the lab is we took these two cell line models to start off. We kind of went on with this SK and B2 cells, which are neuroblastoma cell lines that have more than 10 copies of NMIC amplification. And we also use this 5Y cell line, which is a very common neuronal cell line, neuroblastoma cell line to be precise. In these particular contexts, these cells do not have NMIC amplification, but rather they have c-MYC expression in them. They are less aggressive, and they're not as resistant to many of these first or the standard of care chemotherapeutic drugs. while the BE2, the NMIC amplified cells, are very aggressive and also resistant to a lot of standard of care chemotherapeutic drugs. So to start off, we isolated these exosomes or extracellular vesicles from these cells by optiprep density gradient centrifugation. You can see the fraction 7, where you have the exosome or EV-enriched proteins, ALIX and THG101 in fraction 7. And also you have another population at 9, where you don't have these markers in this particular fraction 8, but we do see them again in 9 and 10. Right, and you can see in transmission electron microscope, you do see some of these vesicles in them. Similarly, the NMIC amplified BE2 cells, you do see also that EV enriched proteins, ALIX and THG101, fraction 7, 8, 9, and 10. But you don't see these two kind of populations as you see in 5Y, but you kind of see a consistent presence of these proteins in the Western blot. But doesn't mean there is a difference in population or it's not heterogeneous, but they are quite heterogeneous as you can see. But the TM is only from fraction 7, but not from all the others. So you can see the vesicles are present, but they're also a bit heterogeneous in terms of their size as well as shape. So we did the proteomic analysis of these exosomes or extracellular vesicles. And as you would see, because now we are doing proteomics on 2 different cell lines, they're different based on various other parameters, not only based on the NMIC amplification status, but also mutation on many other genes as well. So it can be considered as 2 different patients and cannot be completely considered to be dependent on NMIC amplification alone. But however, you can see there are quite a few proteins which are different between these 2 cell-derived exosomes, as one would expect. And we also validated that by some of these proteins by Western blotting, where you can see EV-enriched proteins like ALIX, THG101, CD81 are present in them. Beta-catenin is also present, and also RHO-A as well as NET4. But you can see there are proteins like ALIX and beta-catenin has a small shift in molecular weight in these Western blots, and this is pretty consistent in multiple runs. And we do not know what is the real reason for this shift. It could be some modifications which are present in these proteins, but we are not aware of what those modifications are. When we analyze these proteins by Fundridge tool, what we identified was especially the SK and B2, the NMIC amplified neuroblastoma cell-derived exosomes have a lot of proteins which have been implicated in signaling pathways. Right, you can see all the red ones are those which are abundant in the NMIC amplified cell-derived exosomes, while the blue ones are those which are abundant in the non-NMIC amplified exosomes, as you can see from here, like trail signaling, mTOR, IL-3, and EGF mediated signaling pathways. So with this, we thought, do these exosomes have a functional capability? And then, similar to many other labs, we took these exosomes, isolated these exosomes from the cell lines, used SY5Y, the non-NMIC amplified cells, as a recipient cell, and we incubated incubated on these cells the amount of exosomes for 24 hours, and then we did a colony forming assay. And as you can see here, the untreated cells have some number of colonies here. But when you treat that with SY5Y exosomes, there was increase in the amount of colonies. But once we treat with the B2 or the NMIC amplified exosomes, there is a significant increase in the number of colonies which have been exhibited, suggesting that the exosomes from SK and B2 cells have the potential to increase the colonies that has been formed. Similarly, we also looked at the migration ability, like if the exosomes can induce more migration in these cells. And again, we use the less aggressive SH5Y cells. And when you see the SH5Y cells by themselves, they're not migrating as much. But when we treat them with exosomes from SH5Y, the same cells, there's some increase in the migration. But when you treat that with the BU2 exosomes, you can see a significant jump in the amount of migration that you can see in these cells, right? Suggesting that, yes, the invasive ability of the cancer cells are increased when you treat them with the BU2 cells, and that thereby also conferring the aggressive phenotype of these cancer cells. Till now, what we saw was everything on a cell line base, because now, as I mentioned before, these are two different cell lines, and NMIC is not the only variability in these two cell lines. There are multiple other factors. There's many mutations. Again, the expression of genes and the proteins which are expressed in those cells are varied between these two cell types. So we cannot attribute everything what we see directly to NMIC. So to do that, what we did is we did an NMIC knockdown in these cells using shRNA. As you can see here, this is B2 cells. When you do the shRNA-based NMIC knockdown, you can bring down the expression of NMIC. And then we use these exosomes from the wild-type BE2 as well as the NMIC knockdown cells and treated that with the 5Y cells, which is the Lex aggressive cell, and we did the wound healing assay. And here you can see the untreated cells, the 5Y at 0 hours and 18 hours. And the doubling time is pretty much like 24 hours for these cells. So we did everything less than the doubling time. And we also use the proliferation inhibitor in this particular assay as well. So you can see when we treat the cells with SH5Y exosomes, we can see there is an increase in the wound healing ability. But when you use SK and B2 exosomes, there's a significant increase in the wound healing compared to the control, or even compared to the 5Y cells. But when you also use the NMIC knockdown cell-derived exosomes, we can partially rescue this phenotype, but not fully similar to the 5Y cells or compared to the control. But you can partially rescue the phenotype, suggesting that NMIC has the potential to drive this aggressive phenotype by packaging some of the cargo that may be regulating this particular phenotype. We also went on to show whether this aggressive phenotype can also be seen in terms of treatment resistance. And for this, we used doxorubicin, which is one of the standard of care treatment chemotherapeutic for neuroblastoma patients. And in this particular context, we use 5Y cells, and you can see at the basal rate, this is a cell death without any drug treatment, but with the exosomes treatment. But when you start giving the drug, the doxorubicin, and also with exosomes, you can see the SK and B2 cells that even with 5 microgram concentration, you can see some protection. And it's not as striking when you say 10 and 20, it's not increasing significantly, but there is some kind of protection when you use the SK and B2 exosomes onto the 5Y cells and then treat them with doxorubicin, suggesting that either they can change the pathways by which the cells respond to the apoptosis, or even change the drug efflux mechanism. We don't know the exact reason for why this happens, but there could be multiple potential options for that. But when we use the same way with the NMIC knockdown derived exosomes from the BE2, you can see you can rescue this phenotype where this is the BE2 exosomes, there's the NMIC knockdown exosomes. You can see the difference where the chemoresistant phenotype transfer has been completely abolished when we use these exosomes from NMIC knockdown, suggesting that NMIC has the potential to drive this particular phenotype, either by transferring the aggressiveness or even in terms of the treatment resistance phenotype. So this was again published in JAMA, as I mentioned, and you can go to this particular article and look at it more. But currently we are now doing animal experiments in the same project, but we are using again some of the technologies that many other guys have developed in terms of the Krelok system as well as luciferase system, in terms of seeing how exosomes can be transferred in the tumor microenvironment. But to be honest, the Krelok system has not been as efficient, but the luciferase one is pretty efficient in our hands. So the second story, I'm going to talk about a similar story in this context, another one which we, again, we published end of last year in JEL on colorectal cancer. In this particular context, this has been a study that has been running in the lab for almost like 7 years, I should say. But I have to acknowledge that whatever we tried a lot of those things, but I should say what we end up publishing was somewhere like 40% of what we tried, because 60% of the things that we tried did not work, as is the case in research. But in this context, we thought it could be more robust in terms of the phenotype, but unfortunately, we didn't see that. I'll explain what I mean by that as we go by. But what is this story about is, let's say, as I mentioned in the introduction, when we talk about a tumor, there is a lot of heterogeneity in that. We do have various mutational loads in these cells. So there are some cells in the context of colorectal cancer where there are APC mutated in APC. And there are some cancer cells which are mutated in APC and KRAS. And then you also have some mutations in beta-catenin and KRAS, right? And how do these cancer cells communicate with each other? For example, if you do know that KRAS is mutated and this is gain of function, and can a cancer cell transfer this KRAS via exosomes to another cell where there's no mutation in KRAS, and thereby kind of keeping that proliferative signal longer enough, or even in terms of favor of cancer progression? That's the concept which we started off with. And we mainly focused this particular study on beta-catenin because it's one of driver mutation in colorectal cancer, and also well documented in the literature. And we did have a lot of tools in our hands to study this particular protein. And this was predominantly done by a PhD student, Hina Kalra. She graduated and she's postdocing elsewhere currently. So again, as I mentioned, this is one of the resources we developed called Colorectal Cancer Atlas, because as a lab, we are interested in colorectal cancer. So what we can say clearly is, when you talk about this particular tumor, 93% of genes which are implicated in Wnt signaling pathway have been known to be mutated in this particular tumor, right? And also, in a way, like when you talk about these individual genes, like there are 16 different Wnt signaling pathway genes that have been mutated. Most importantly, APC, that has been known to be mutated in at least 81% of the cancer patients. And then you have Frizzle receptor, as well as the beta-catenin, which is also mutated in 5% of the cancer patients. So when you talk about Wnt signaling, when there's no Wnt ligand, normally what happens is you have beta-catenin there, which is phosphorylated by this destruction complex, and then it targets the phosphorylated beta-catenin for degradation. In cases where there is Wnt ligand, now this destruction complex has been inhibited, and now beta-catenin is free and cannot be phosphorylated, and this kind of beta-catenin translocates to the nucleus and then binds with this TCF and then activates Wnt target genes. And this is what we see in a lot of cancers, including colorectal cancer, where do we see mutations in APC and beta-catenin. So now, in our lab, we, as Sherry mentioned, we do use bioinformatics and proteomics quite often. So what we ended up doing is we did exome sequencing of quite a few cell lines that we had in the lab. And we used this LIM1215 colorectal cancer cell line, which is derived by this Ludwig Institute in Melbourne. So when we did this exome sequencing, what it precisely gives is the mutations in the coding regions in this particular cell line. So then we translated these mutations into proteins, because when you do mass spec, and when you're trying to identify the proteins, mass spec will not identify a mutation if that particular protein mutation is not present in the database that you're searching. So if you create a translated mutant protein database, then when you search your proteomic data against them, you may end up finding the mutant proteins. And that's the approach we took, the proteogenomic approach. And we isolated exosomes, we did proteomics on them, and then we searched that proteomic data against a mutant database, which was derived from the exome sequencing data. And then now we were able to identify many mutant proteins that are secreted via exosomes in this particular way. So what was interesting in this particular observation was high levels of mutant beta-catenin was secreted via exosomes. And we focused mainly on beta-catenin because, as I said earlier, we were looking at driver mutations and those which will result in gain of function. Meaning if a particular mutation results in a gain of function, it can also go to a recipient cell, and then that gain of function can turn into an activity. And that's why we focused mainly on beta-catenin. For those who are interested, you can see the exact mutation we see in beta-catenin is pretty much in this exon 3, A121 to G. And what it translates in the protein is where you have a serine residue at 43, and which is supposed to be phosphorylated, and now it's converted into an alanine, which cannot be phosphorylated anymore. And that kind of renders beta-catenin to be not phosphorylated by the destruction complex, and thereby always constitutively active. So again, to validate this particular observation, we took the exosomes and did Western blotting. And you can see beta-catenin is found in the exosomes secreted. Again, this is a crude prep of 100,000 G. Then we also looked at the soluble secretome, which is like the conditioned media without the extracellular vesicles. We can see it's not present in the conditioned media, but present in both exosomes and also in the 10K fraction, which have a little bit of beta-catenin in them as well. Then we floated them in OptiPrep density gradient centrifugation to see that whether they also kind of pellet at the same density as the EV-enriched markers like ALIX and TSG101. We can see beta-catenin is similar to TSG101, suggesting that they are indeed associated or present in the exosomes. But based on various other analysis, we do think beta-catenin is present inside the extracellular vesicles. So as I mentioned before, in spite of whether there is any Wnt ligand or not, if there is a wild type beta-catenin, it results in degradation. But if there is a mutant beta-catenin, whether there is a Wnt ligand present or not, there will always be an activation of Wnt signaling pathway, where beta-catenin is always localized to the nucleus, and then it activates the Wnt transcription genes, right? And to confirm that, we use these two cell line models, as I mentioned. One is LIM1215, which is the Ludwig Institute Melbourne 1215 cells, and then the RKO cells. So the LIM1215 cells have a mutation in beta-catenin. As I mentioned, this is the serine residue, which is in the 43rd position, and thereby it cannot be phosphorylated. And here you can see a confocal microscopy image of beta-catenin, which is in green, and the topro is for the nucleus. And you can see beta-catenin is predominantly present in the nucleus in these cells, right? And in the other colorectal cancer cell, RKO cell, there's no beta-catenin mutation. It's pretty much a wild type. You can see it's more often present in the cytoplasm, and it's not predominantly found in the nucleus as you see in the LIM1215 cells, right? This kind of goes on with the literature saying that if there is a constitutive mutation in beta-catenin, it normally goes to the nucleus. But however, we also need to validate whether the beta-catenin is functional in the nucleus. Does it bind to TCF? Does it activate the Wnt signaling pathway? And for that, we kind of did a luciferase assay. So the luciferase assay, you can see there is pretty much almost like a 2,500-fold increase in the Wnt activity in the LIM1215 cells where there is a mutation, but in the RKO cells, it's pretty much basal, and there's not much significance in there. So what is this luciferase assay, as I mentioned before, because this is nothing but very, very specific to the nuclear beta-catenin. So you have the TCF binding sites for beta-catenin in these particular plasmids. And when you have beta-catenin binding to them, this will express luciferase. But there is also a negative construct of FOP where the TCF binding site is mutated. So because of that, beta-catenin cannot bind to that. And we also have a negative control whereby the FOP, the mutant sites, will not elicit the luciferase signal, right? And based on that, we get the top signal, which is the positive signal when it binds to TCF. and the FOP signal, which is the mutant consent, so the negative signal. So based on that, we can get a particular signal for the nuclear activity. And this is very, very specific to beta-catenin. No other gene have the function in terms of no other protein we can attribute the function to this luciferase assay. So now we asked a simple question, as I mentioned before: can the mutant beta-catenin transfer via exosomes and elicit the Wnt signaling activity on the recipient cells? Right, as one would expect, because these beta-catenin, as I said, these are constitutively active. You express or overexpress them into any cell, you will have a Wnt activity in them, as we all know in the literature. Similarly, when we took 30 micrograms per mil of EVs and then incubated for 24 hours on LIM1215 or RKO cells, we can see induction of this luciferase activity in those recipient cells, suggesting that the Wnt activity is going up. So is the upregulation of the Wnt target genes. Like, say, you can see c-MYC is a well-known Wnt target gene. You can see the expression goes in a dose-dependent way when you put more amount of exosomes. And you can also see the beta-catenin expression goes a little bit higher in a dose-dependent way in these cells. We also looked at phospho-AKT activity, which was not changing, but there is in the literature, it is known that phospho-AKT controls a little bit of beta-catenin signaling as well, but we couldn't see that in this particular cells in our hands. So we also validated the upregulation of the Wnt target genes like c-MYC and cyclin D1 by PCR as well. In addition to Western blotting, you can see that with 20 and 150 micrograms, you have the RNA expression going up in these cells, suggesting that not only the luciferase activity, also the Wnt target genes are also going up when you incubate the EVs onto the recipient cells. So now, one thing I need to make clear is, because now we are taking exosomes, 30 micrograms per mil, and this is, beta-catenin is not the only protein that is present in them. There's a lot of proteins, a lot of RNAs which are present in them. How confident are we to attribute that to beta-catenin? Right? This could be a function that could be regulated by thousands of proteins or genes which are present in them. So to be putting the blame on one particular protein is not always right. So for that, in this scenario, we do know that the luciferase assay that we performed is very specific to nuclear beta-catenin. So the function that we saw in terms of the luciferase activity can be triggered by two different phenotypes. It could be either by the mutant beta-catenin that is transferred via the exosomes to the recipient cells, or it could be also by Wnt ligands, which can be transferred via exosomes, right? And we do know that Wnt ligands are secreted via exosomes. Like, a lot of people have shown this from 2012 onwards, where Gross and Luga et al. Have shown that various Wnt ligands can be attached to the membrane of the exosomes or extracellular vesicles and can be transported. In our hands, in our setting, this could also be possible, but we haven't done anything to rule it out, because the ways we can rule this out is either by checking for various Wnt ligands. The antibodies for many of those Wnt ligands are not that great. Another option is trying to knock out beta-catenin in these recipient cells by CRISPR-Cas9. And we did do that, but once we knock out beta-catenin, the cells become senescent and they don't take up exosomes anymore. So it goes into a different phenotype when we try to do, and we are not able to prove whether it's only beta-catenin or Wnt ligand. So in this context, we are saying it could be possible that both these things can work in tandem, but we have proof that beta-catenin is being transferred to the recipient cells. So how do we confirm that the beta-catenin is going into the recipient cells? For that, we adopted this SILAC approach. Again, SILAC is stable isotope labeling of amino acids in cell culture. So now you have, let's say, you grow these cells in normal media, the LIM1215 colorectal cancer cells were grown in normal media. And while the RKO cells, the recipient cells, were grown in heavy media. The heavy media, again, we kind of play with the nitrogen carbon composition of the amino acids in these cells. And now we can see after 9 cell doublings, because we need to make sure that every protein in that particular cell has incorporated heavy labeled amino acid in them. And once we confirm that, we kind of took exosomes from the LIM1215 cells. And in this case, I apologize, it's not 30 micrograms, it's 150 micrograms per ml. 30 was not enough because we have to use a very high dosage because for us to get any amount of protein in the nucleus, we had to use 150 micrograms per ml to get any signal in them. So we transferred exosomes onto the recipient cells and then waited for 24 hours and then took the RKO cells, washed them thoroughly, and then did subcellular fractionation, meaning we took the cytoplasmic fraction, nuclear fraction, the whole cell lysates separately, and subjected them to Western blotting and mass spec. So now, why we did this is like, to be honest, like when we find any heavy labeled peptides in a recipient cell, we do know that is all coming from the RKO cells, which is being made endogenously by the RKO cells. But if you pick any light peptides, these light peptides or from normal amino acids are coming from the exosomes per se and not made by the RKO cells. So we do know for confidently that these are transferred directly from exosomes, and thereby we can kind of see what proteins are transferred and whether they go into the nucleus or they stay in the cytoplasm, things like that. So again, this is a validation of the subcellular fractionation. You can see the nuclear fraction is pretty much enriched with the nuclear proteins, the topo 2b. Samely, you can see the beta-catenin is mostly present in the cytoplasmic, and suggesting that our preparation is pretty good. So then we subjected these fractions to mass spec, and what we found out was, as expected, we could see two peptides, light peptides were picked for beta-catenin, and this is the peptide, and this is the MSMS, tandem MSMS spectra, which you can see here. And these were picked exactly in the nuclear fraction after 24 hours, right? And suggesting that we do see transfer of beta-catenin, mutant beta-catenin to the nucleus of the recipient cells. We also have done confocal microscopy to show that there's an increase in signal of beta-catenin when you see that in the nucleus. Most importantly, what we also found was, apart from beta-catenin, we saw several other proteins, including histones, many methyl transferases being transferred to the nucleus and the cytoplasm of the recipient cells. And some of these findings we are following it through. For example, some of these histones, again, even though the histones are also present in the recipient cell, why would these histones go to being transferred by exosomes? Is there any function for them? That's what we are following up. And the list of all these proteins that we find in the nucleus can all be found in the supplementary table in the publication that has been uploaded last year in JEM. So pretty much now we did everything in vitro, right? So now we need to move on to in vivo, and we wanted to see whether what we see is reproducible in vivo. And this is where we spent almost like, as I mentioned before, almost like a lot of effort, like many years of work went on to it. But unfortunately, we were only able to publish a little bit, but many things that we tried did not work. Right? And one of the things that we worked, I'm going to present here, is where we took the RKO CRC cells, made a mouse xenograft of that. And then we also took the exosomes, labeled it with the lipophilic dye DIR, and now injected these exosomes intravenously into these mice. And the questions that we ask here is can we pick up these exosomes in the mice at various time points? And also, if so, what happens to Wnt signaling activity in this mice, whether the Wnt target genes changes by qPCR or Western blotting in the tumor tissue as well as other tissues, right? So this is first experiment that we did, and after we followed the in vitro one, because we thought it's a straightforward experiment to start off with. As expected, you can see at 24 and 96 hours, the biodistribution, which is now pretty common, where you can see when you label these exosomes, you can see the tumor control is not lighting up. You can see the treated tumor cells do receive these exosomes in them. Lungs always receive a lot of these extracellular vesicles because of the intravenous delivery. And also we do see a lot of these vesicles in liver, kidney, and GI tract as well, as you can see in this particular picture. So yes, we can see these EVs, but what about the Wnt activity, right? So for that, again, we also wanted to make sure that we also wanted to have make observations of the tumor, as well as look at what happens to this tumor at various time points. And for that, we did the same experiment with the RKO cells, and we also used 10 micrograms of EVs. So why did we decide on the 10 microgram again? This, when we did, it was a long time ago, and we equated this on two particular factors. One was we did have David Leiden's pre-metastatic paper at that time, where they also used 10 micrograms of EVs in the mice, and then we tried to follow it up. And the second reason why we did was when we do established a xenograft mice with LIM1215, and we isolate the plasma from those mice, and we do get somewhere to 5 to 10 micrograms of exosomes in addition to a normal mice. So to put that in other words, if I take the plasma of a wild type mice and plasma of a xenograft mice, and we calculated the amount of exosomes or EVs that are present in those mice, and we can see somewhere like 5 to 10 micrograms of increase in those mice at one time point. But whether this particular concentration equates that to the mice is a different question, because 5 to 10 is what we are seeing constitutively. At any time point you take the plasma, you have 5 to 10, but this is a one bolus concentration of injection, so which may not directly reflect to the concentration that we see in the physiological conditions. So we started off by doing this because when we tried 30 or 40 micrograms in some of these mice, the mice died by coagulation. So we didn't want to give a lot of exosomes into them. That's why we started off with 10. And to be honest, we are not sure whether this is physiologically relevant, but we started off using that in a way. So what we saw was, again, when we treated the mice with and without exosomes, these are LIM1215 exosomes, which contains the mutant beta-catenin. You can see the Wnt target gene c-MYC goes up in the tumor tissue of the treated mice, right? Similar to what we saw in vitro. Similarly, you can also see the mRNA expression also goes up, and AXIN2, which is also a Wnt target gene, also goes up in those treated mice tissues, suggesting that Wnt activity is increased when you inject the LIM1215 exosomes containing the mutant beta-catenin. What we also see is, when we also inject these exosomes into those mice, we also see increase in the tumor burden, right? And again, whether this is only because of beta-catenin, it cannot be, because there are many other proteins and RNA in those EVs. So we kind of think it's a collected effect in terms of, so we cannot attribute the increase in tumor burden just because of beta-catenin. But we do know that this kind of increase, especially in the Wnt activity, could be attributed to beta-catenin, but not the wholesome tumor increase, right? So that's pretty much where we ended the story. But we also tried a lot of things which didn't work. I just want to mention some of those things. One is the Trelock system, where we wanted to, that was published by Jacob van Reenen in a Cell, where they used the horizontal transfer system, where the exosomes will have a Cre recombinase being secreted out, and the recipient cell, when they fuse with the recipient cell, the exosomes, and then they can kind of create this recombination event and kind of change the color from RFP to GFP, and then or GFP to RFP, depending on how you set up the cells. And based on that, you can kind of say whether the exosomes are transferred or not. But unfortunately, the amount of Cre that goes into the exosomes is very, very low. They do not get directly packaged into the EVs. But if you modify them by putting some targeted linkers, then you can get more of them packaged into the EVs. But we tried this for a few months and years, but we didn't get it to work. So I had to drop it. As well as we went on to do some patient-based samples and try to identify whether we can find beta-catenin containing exosomes in there. Again, there's we do see beta-catenin, mutant beta-catenin in the plasma of the patients, definitely in them. But how much, whether they are physiologically, whether they can induce the Wnt signaling activity, we haven't tried in our hands. But yeah, so in a way, other things that we tried also knocking out beta-catenin in these cells, and again, that didn't go as well as we planned. We had a lot of difficulties in this particular project, but finally we ended up packaging it out because of the time limit as well. But nevertheless, what it kind of made us learn here is you need to be very, very careful, especially in terms of looking at the functional aspects of EV. Some of them, what we are studying, may not be physiologically relevant. We are putting 30 micrograms, 20 micrograms in vitro conditions. How much they are relevant physiologically, nobody knows. But it's a good way to start and go on to do it. The best way to do is always use a dose or a titration curve where you can go very low amount of EVs as well as high amounts, and then you can scale it down, right? But anyway, so I'll stop off because of the time. So I'll stop off with acknowledging the people who have been involved. This is the lab in 2020. Again, I think some people are working in the lab, or maybe 2 or 3 of them are working in the lab, rest of them are still at home. Most of the work was done by Kamali and Hina, as I mentioned before, and also acknowledge the funding agencies and also a lot of collaborators who have been involved over the years. And also thank you for your attention, and happy to share any reagents if you want any of us. Thank you.
Super. Thank you, Suresh. That was awesome. And I'm really pleased that you were really honest going, yeah, things don't work. Because we nearly tried that Crelox system. I'm quite pleased we didn't, because you've gone through the pain for us. So has anyone on our Zoom call got any questions? How's Wellington doing for questions? Very quiet.
Oh, sorry, we're just getting our technology working again. That was really interesting. Thank you, Suresh.
Thank you.
So I was just wondering, the work that you did with the exome sequencing and then the exosome proteomics, was that all just with the LIM1215 cells, or did you look at other cell lines as well?
Yeah, we did look at 7 cell lines.
Oh, wow.
And we did exosome proteomics on all of these 7 cell lines. And why we chose LIM1215, because of this beta-catenin mutation in them. Because as I mentioned before, 5% of the cells have beta-catenin mutation. And we did see this mutation in beta-catenin. And because of the strong effect of the cell line, we chose that and didn't pursue the others.
Well, that's really interesting. Do you guys have questions?
Wellington's shy.
Huh?
Wellington, this is like the Eurovision Song Contest. Going over to Wellington. So Auckland, any questions from Auckland here? Everyone's very quiet. I was interested in your histones, and I think Matt would be interested in your histones, and about identifying your histones being transferred. Did you say that they went into the nucleus?
Yes. So we do know that they are going into the nucleus, but what we don't know is whether any part of DNA is present in them. Because histones bind to DNA, as we know, and whether any of these histones are there to preserve those DNA from degradation, we don't know. But we do know for sure that they are going into the nucleus. Some of these histones are acetylated as well at certain spots, and we don't know the functional relevance of why they are acetylated, right? And pretty much all the histones that we see, we could also pick those histones in the recipient cells as well. So in a way, we do not exactly know. But another study that I would probably mention on that is like we also did the methylation screen in terms of looking at the recipient cell and to see whether there is any change in the methylation status of the cells upon incubation of exosomes. And we did see a lot of genes being either hypermethylated or hypomethylated, but it was not a striking difference between the two cell lines. For example, if I take a resistant cell line and a parental cell line, you can see the methylation pattern is quite striking. But when you put an exosomes on to a recipient cell, there is changes in some of the genes, but it's not striking per se. So there's definitely some methylation happening, and histones could be also regulating some of the acetylations in them. And then, yeah, we don't.
Sorry, I have another question based on that. Was there methylation of anything like MLH1 or similar genes that are associated with microsatellite instability phenotypes in people?
To be honest, I don't know the exact name. We didn't see about the microsatellite instability genes, but what genes we found out was some of the cell adhesion genes were methylated and they were silenced. And then there were also some genes which are pro-apoptotic that were silenced. And there was also some anti-apoptotic genes which are silenced. So we kind of try to validate this based on by doing some bisulfite sequencing, and we didn't have the capability to do that. But we kind of report it as a whole genome sequencing based exercise, but we haven't validated them per se, so that we haven't followed it up much.
So beautiful talk. Thank you very much. Can you tell us how long the effects that you're seeing last? So when you're transferring beta-catenin mutants into cells in vitro, or even those methylation effects, are they carried through generations, or do you need to keep adding fresh exosomes to maintain whichever of those activities it is? So is this a permanent change or a temporary change?
Yeah, so all the changes that we see from the first part of the talk, where we use neuroblastoma cells for migration, or even in terms of chemo resistance, or even in terms of beta-catenin, Wnt activity, everything is temporary in this particular exercise. But as I mentioned before, as part of this study, what we ended up doing is we wanted to see whether beta-catenin is driving everything. So we did end up doing a beta-catenin knockout using CRISPR. And when we did beta-catenin knockout, the cells became senescent, right? And when we took the exosomes from those senescent cells and put it onto a recipient cell, they became senescent immediately in 16 hours. And that was almost permanent. So the phenotype that we see in this particular talk is all temporary, but there are some phenotype which are transferred are permanent, like example senescence. So in other words, we have to keep supplementing more exosomes to see this phenotype continuously. But in physiological conditions, you would expect to see that, because these exosomes are constitutively secreted all the time while we are giving a bolus concentration of just one treatment. So you would expect that the function is going to be retained, and this particular tumor favoring functionality is going to be retained because of the constitutive secretion inside physiological conditions.
So can I just check that I got that right? So you were able to transfer senescence rapidly with vesicles?
Yes, within 16 hours. When you co-culture the cells that are senescing and exosomes from those senescent cells, when you transfer them onto the recipient cell, in 16 hours, they're able to transfer the senescence. And this is not, yes, we did see this 2-3 years ago, but right now there are 2 papers already confirming this. One is from Anna Olen's group, which is published in Cell Reports. You can see that, which came last year. And there's a paper in Cell Metabolism, which came just a week ago, which also confirms the GTPase activity for exosomes from senescent cells. Definitely, there are some which are permanent and then some which are not so, depending on the context.
Oh, thank you very much. Any other questions on Zoom? Lots of quiet people. I've already picked on Wellington.
I have a question.
Go for it, you hear me?
Hi. Yes, have you looked at cancer-associated fibroblasts and the influence of their exosome, because they are quite they're sitting in the same niche.
That's right. So we did look at cancer-associated fibroblast and look at it. So if I have to, one second, I'll just. So we did look at all the cells in those tumor microenvironment that I mentioned in terms of cancer-associated CAPS, the endothelial cells, as well as the, I just can show this. But we didn't specifically focus on cancer-associated fibroblasts mainly because there's a lot of literature on CAPS in the context of the tumor microenvironment. So we focused mainly on the endothelial cells and the endothelial cells precisely. And what we are able to see that is when you take exosomes from the cancer cells and treat it on the endothelial cells or treat it on, let's say, the cancer-associated fibroblast, we took the exosomes again from the treated endothelial and CAPS and treated back onto the cancer cells and saw whether that is favoring cancer progression or not. And we did see both CAPS and endothelial cells were facilitating cancer progression, but they needed exosomes from the cancer cells first to go to them and educate them, and then whatever comes back to them in a reciprocal way, then kind of favors cancer progression. But as I mentioned before, we did not focus a lot on CAPS because a lot of people working on CAPS. So we mainly worked on pericytes, endothelial cells, and other immune cells, the NK cells and the macrophages in this particular context.
Thank you. Because I thought that the CAPS actually influence Wnt signaling as well.
That's right. And one of my collaborators has a lot of CAPS in his hands, and he keeps kind of bothering me saying that to work on CAPS all the time. Anytime I email, he's like, what about CAPS? Can I send CAPS? So probably I may need to enter into that, but we haven't looked at it, to be honest. There are a lot of people, like, for example, Oliver from Belgium, and again, quite a few people have shown CAPS have a lot of role in this particular phenotype.
Thank you.
That's okay.
So any more questions? Any less shy people on Zoom? Everyone in Auckland's quiet, and Wellington looks like they're hungry. They're probably hungry. Okay, so I think we'll leave it there. Thank you so much for joining us, Suresh, remotely. Next time, I promise, we'll get you on a plane and you'll be able to come over and visit us. And we'll get you jumping off the Sky Tower like we nearly did with Andy.
Are you sure even the Australian states are not letting us in anymore?
Yeah, well,
we are banned.
We've got taste. We're going to ban you guys. Yeah.
Victorians are banned within Australia itself. Forget about it. Yeah.
So thank you so much. Thank you. I'm just going to give a clap because I'm used to giving a clap.
Thank you guys. Thanks for your time. And yeah, we'll definitely have more collaborations within New Zealand, definitely.
Definitely. We'll be in touch by email. In fact, we also want to know what antibody you're using for Alex because we can't get ours working.
Oh, okay. So yeah, we can share it. It works pretty well.
Your Western blots look much nicer than ours at the moment, so we might be in touch as well.
So our Alex works like beta-actin because anytime we kind of throw it along with TSG101 as well because they work beautifully in any time you throw them.
Yeah.
And these antibodies can be reused very well as well, especially Alex. Yeah.
Yeah, we're doing well with CD81. That's our favorite one at the moment, but the others are a bit of a pain.
Yeah, okay.
Right, I'm going to leave it there because I'm just chit-chatting. Thank you for joining us. Thank you, everybody. We will reconvene in 2 weeks. Are we still looking for a volunteer for Journal Club in 2 weeks' time? You don't have to be talking about a journal paper. You can talk about your own research. And we'll also be trying to get more people online via Zoom to join us as well for presentations like this. Thanks, everybody. Bye.
Thank you, guys. Thank you all. Bye. Yeah.